Hey everybody, Mr. Boyd here. We are going to talk about sponges today. And this will be your second video of the week. Your first video was an overview of Kingdom Animalia, general features, symmetry as an idea, an overview of the diversity of phyla of invertebrates. And now we're getting down into our first true phylum of invertebrates, the poriferans, pore-bearing creatures. These are the sponges. And so my plan, I'm thinking 3 video lectures a week between 30 and 40 minutes. I'm trying to get a sense for how much is a good load. So bear with me. I appreciate your feedback today. I know it was difficult for a couple of you guys to see some assignments. And I think some of that was my issue with the settings, not having them right. And so my apologies for that. Again, unless there's a due date that's stated, you're not, or rather a submission, you don't have to tell me you've read the chapter. I'm going to assume you are doing all of your assignments, so the trust is in your court. And then I'm going to have some assignments that will have a follow-up study guide questions as an example. And so you would submit those. You can submit those via LMS on Facts as a photo, as a Word doc, if you want to just write your answers in Word and submit it that way, or you can scan them in. Or if you'd rather do it the hard copy way, my box is there in the cafeteria, and you have until the following Monday to get everything in. So you'll see some due dates, and that's to keep you on track. I think it's going to be more helpful to just spend an hour a day doing bio and getting those things done each day. But it is open through the week as Mr. Whitling's email suggested. And so you'll be able to submit those before the following Monday. Let's pray. Lord, thank you for this day. Thank you for these students. Thank you for your wild creatures and these marine creatures that we get to study a little bit more in depth. And I pray that it would be a joy. In Jesus' name, amen. So Phylum Porifera, these are the sponges, guys. And a wild world of sponges. We are still learning a lot about sponges. Lots more to know. A refresher on characteristics of Kingdom Animalia. They are eukaryotic, multicellular with cell differentiation, cells that lack cell walls, heterotrophic creatures, and motile at some point. Motile, of course, being the opposite of sessile. Sessile, S-E-S-S-I-L-E. And you're going to find out here early on that a few of these intro phyla have stages of their life where they're one or the other. So that's kind of interesting. This is a favorite of mine. You will greatly enjoy watching the sponge video with Jonathan Bird. He's a character. I might have shown him in class once. He's definitely an evolutionist, but he loves the marine environment and the creatures there. And he does a pretty good job of really an amazing job of videography and a really decent job of discussing the creatures regarding their characteristics, anatomy, etc. So enjoy that. There's also a historical bit there, getting back to sponges being used regularly in the bathrooms as a sponge for scrubbing dishes, for scrubbing in the tub. Here's a barrel sponge, this massive sponge that someone could fit inside of. This is an animal. This is an actual animal. And you're going to learn about how and why this is an animal. It's obviously sessile now as an adult. So I want you to be thinking about some things that we've talked about already. We've talked about symmetry in particular. And how is symmetry going to work with these creatures? And maybe you've seen one that looks more refined like this. The old bath sponge, really nice and soft. You can buy these still today. They're crazy expensive now, but they're beautiful. They're really a neat, and that's the leftover skeleton, quote unquote, the protein spongin that you have right there. And you'll find out soon why sponges are not created equally. You don't want any sponge to bathe with. If you pick the wrong type, you're going to just get scraped up really good. And so a little bit of a refresher and just touching back on that slide where I listed all the invertebrate phyla that we are looking at and their diversity. And I listed 5,000 species. I found nothing but conflict in trying to pin down a number of sponge species out there. The most reliable source I could find said 6,000. Lots of them say 5 to 10,000, which is just not acceptable. What kind of range is that? And so others say 5, 6 sounded good. And a lot of times, some of that range, which I couldn't find details about, include many extinct species. So we know there are extinct. Almost all of these phyla were found in the Cambrian explosion. If you remember the Cambrian explosion, this is when we found the diversity of life forms all arising at the same time in these silos in this orchard of life. And we still are discovering a lot about sponges today, especially in colder waters. We kind of thought of sponges as really warm marine creatures for a long time, but we're discovering more and more in temperate waters in the North Atlantic and North Pacific in particular. So porifera means sponge-bearing. They have specialized cells, and this is interesting here, and this kind of fits with what we've studied so far. These creatures have specialized cells, but they don't have true tissues. They don't have muscle tissue. They don't have nerve tissue. They don't have a skeleton. They don't have a true skeleton. They don't have any body system you can think of. They don't really have a digestive system, a respiratory system. These are remarkable multi-purpose cells that are functioning and doing multiple tasks at the same time. And so they happen to be, because they don't have, in large part because they don't have these integrated systems, they don't need symmetry. They can function just fine without symmetry as a tool for organizing complexity. And so we see a wide range of sponge forms. Some are more amorphous than others, but all of them typified as kind of this vertical growing, typically hard type of structure. Parrotfish eat them. These parrotfish with these beaks in tropical waters will just crunch on these sponges, and then their feces will mostly be made of sand. And so they're actually replenishing the sand supply in the ocean as they carry out the task the Lord has given them. Lots of beautiful sponges out there. This is a glass sponge. This group is very interesting to me. I will mention an expedition that there's this ship called the Okeanos Explorer, and I'll show you the website of the Okeanos Explorer. It does at least 1 or 2 big trips a year. I was able to have my marine biology students tune in for a deep sea submersible dive where we were watching in real time what they were exploring. And they're exploring just off the coast of Rhode Island and Nova Scotia in the North Atlantic Ocean, places that had never been explored before. So much of the seafloor is unexplored. And so our knowledge of, especially temperate waters that are deeper, really our understanding of the creatures there is growing rapidly right now. So general characteristics of this group, Phylum Porifera. And this goes along with chapter 21 in your text. They are sessile as adults. They are locked down. They glue themselves to the seafloor. The larva, as many marine creatures, many marine invertebrates have this similar type of strategy where they're motile as larvae and they settle on a hard substrate. They need a hard substrate to latch onto, and then they'll grow into a mature form. And you can see the mature, this is a certain species of glass sponge right here. Glass sponge, you'll find out why in a moment. I did want to mention that there are freshwater sponge species, but out of about 6,000 sponges, only 150. So the vast majority are, and I haven't looked into our region. It'd be interesting to find out if we have sponges. I know there are certain rivers in the eastern U.S. that have sponges, but I don't know about the Intermountain West. I'll have to find that out. So you can kind of tell a sponge when you see it. This looks a lot like a sponge. This looks a lot like a sponge. This one could pass for coral if you didn't know, right? But that big opening in the top is a giveaway. Those big openings in the top. And this has to do with how they respire. They don't have a true respiratory system, but they do get rid of waste gases because they're carrying on cellular respiration. They need oxygen, they need glucose, and they need those for cellular respiration, and then they'll produce carbon dioxide and water. All right, and so the way this works is there are pores, ostia plural, ostium singular. There are pores in the sides of sponges, and you'll see this in the Jonathan Bird video. He does a wonderful demo showing how the water flow works. So want to think about this as a cost benefit. If you're a sessile creature and you need oxygen and you need food, what in the world are you going to do? Well, you need a system for drawing those to you, right? And so these creatures have pores, and they have these amazing cells, which are equipped with flagella that whip back and forth and actually draw the water through the pores. Oxygen is exchanged, little bits of detritus and plankton are extracted, filtered out, and then moved and digested, and then the water goes out the top. The top we call the apical opening. Apical means top, and that's called the osculum. All right, so in through the lateral pores, out through the osculum. And you get a nice little flow here. And so just goes to show you they really kind of create their own ecosystem of sorts here with their own little micro currents, etc. So pores are lateral, osculum is apical. And we're going to look at some of these specialized cells that are shown here on the right with that zoomed in view. This image and a couple of the other ones here are from the CK12 Foundation. Check them out. They're a really awesome resource, and I believe it's free to use them right now. So if you want more info, they're a good source. And so let's look at some of these details now, starting with spicules. You can see that spicules has an arrow drawn to it. That's a fun term, a spicule. And it sounds kind of sharp and jagged, like a spike. Well, that's because that's what it is. And this is what makes up the rigid outer layer of sponges, quote unquote skeleton. It's not one uniform mineralized structure. It's many, many thousands and millions of these strangely shaped spicules. Some are made of calcium carbonate. Some are made of silica. That's why they're called glass sponges. Silicon dioxide, SiO2, that's glass. And many, many invertebrates in the marine environment are made of one or the other. At least part of their bodies are. And so we want to start thinking of seawater as this rich soup of different compounds that can be extracted and used. So these creatures are able to extract that calcium carbonate or silica out of the marine water and form these spicules. And look at the diversity of them down here. And these are just different species. They're remarkable. This one looks like kind of like a double harpoon. This one looks like pollen. I mean, there's some wild shapes here. This one looks like some type of grappling hook. This one looks like a kind of like a wind turbine. Amazing. So those are spicules, guys. And here we see some under a microscopic view of them. And I've been able to buy just a small sponge from, say, Carolina Biological Supply and actually get spicules out of it and look at them under the microscope. So this is something you can do if you are curious and interested, getting a sponge and tearing it apart and looking at it in detail. Quite enjoyable. Okay, here's another slide of spicules. And you can see some of the similar types of shapes over here on the left. And this is in an intact sponge. And there's a nice scale for us, about 500 micrometers. So these are small, guys. But easily visible under the microscope, compound light microscope. So most sponges are made of spicules and this special protein called spongin. You might have known as with a lot of our other macromolecules, we have certain suffixes that indicate who we're talking about. So OSE, glucose, lactose, that's a sugar. If we have something that ends in ASE, it tends to be an enzyme. If we have something that ends in IN or EN, it often is a protein. So collagen is an important protein found in bones and other tissue. And this is called spongin. Really, really clever name here. And you can see that this sponge over here on the right is one that definitely has a lot of spongin in it. You can see it's been combed and prepped for sale for use in the bath. You don't want a whole bunch of spicules there. So some are more of one than the other, and some are just kind of exclusively one or the other. So spongin is this tangle of protein fibers. So protein is more than just a steak, right? It takes a lot of diverse forms. Why? Because the DNA codes for a gazillion different types of protein. Remember, that's the end goal of the central dogma, right? DNA to RNA to protein, transcription and translation. All right, back to our diagram again, and this time we're going to go take a close-up look at the choanocyte. So two really important specialized cells. We don't have specialized tissues, but we have all these specialized cells that are kind of in this matrix that work together. And the three are the spicules, the choanocytes, or collar cells, which we'll look at now, and then amoebocytes. All right, here are the choanocytes. Amazing little cells that have a variety of functions. These really are multi-purpose cells. And here you can see the choanocyte. There's the cell body. They have a collar. They have a flagellum. And they're whipping back and forth. And all of the gazillions of choanocytes, flagella, are moving, drawing water through those lateral pores so that oxygen can be extracted and food particles can be trapped. And so the collar traps food particles. And those are then digested by another important cell we'll talk about in a minute. So that's the job of the choanocyte. Move those flagella, cause the little microcurrents to occur. That brings in oxygen, that brings in nutrients, that brings in detritus, that brings in plankton, all kinds of stuff that can be filtered out and then digested. So we have spicules, we have choanocytes, and then we have amoebocytes. And amoeba, I found out either spelling is acceptable apparently now. You can have the O in there after the E or not. And so I see both. I've seen the other one more commonly, but this site used just the E. So the amoebocyte. And you can see the amoebocyte. And also, I didn't mention this. Look at the location of the flagellum in these choanocytes. The flagella is oriented to the outside, right? That is kind of the epithelial layer, the outer layer. Outer is always epi. Meso is always middle, right? So we often have epi and then meso and then some endo layer in some of these creatures. I'm not going to require you guys to know that. But do recognize that the flagella is oriented that direction, which makes sense because it's creating those currents. And the little green blobs that are moving, these are cells that are moving all around. The choanocytes are really fixed in place. The amoebocytes are not. And we know amoebas. We know how they work. We've watched the amoeba horror flick a couple times. And they are little blobs that have pseudopods, right? And so cytoplasmic streaming, they extend those pseudopods and they're able to grab and digest food and nutrients and transport them to various parts of the creature. They produce the gametes. Listen to this list. These guys are overworked. They also gather and get rid of waste. They secrete spicules and they repair damage. And so without a specialized tissue in place, these creatures rely on these specialized cells with remarkable capabilities. This is the biggest list I remember seeing really for any type of cell, with the exception of unicellular organisms. We know that unicellular organisms, by default, they have to do everything, and so they truly are remarkably. And in that sense, the smaller the organism and the fewer cells, in a way, they're more complex. They often can breed multiple different ways, and they have to carry out all these tasks that in us and quote unquote higher animals, we have complete organ systems that do those tasks. Okay, so asymmetrical, no true tissues, no circulatory, respiratory systems. Let's talk a little bit about their reproduction here, and then we'll be done. Here's a cross section of a typical sponge, and I wanted to include this one to just point out some of the vocab terms that your author has. He has a couple of excellent diagrams. You can see the choanocytes on page 261. You can see the spongiocoel and the radiocanals on page 260. So do take note of those. And here we see the spongiocoel. This is the inner cavity. That term C-O-E-L means cavity, seal, spongiocoel. We see the choanocytes, and these choanocytes are on the inside. And so I haven't checked I haven't fact checked that positioning. It's possible that they line the inside as well, or maybe in some species they're found on the inside and not on the outside. And then the osculum, the apical opening, and then another item I wanted to point out here to you guys are the radial canals. And the radial canals aren't really shown here. You do see these pores, but there are radial canals found in these sponges, and please notice them in your textbook. Okay, so these radial canals are on page 260. So again, water moving through the pores that are lateral and apical out the osculum. Reproduction in sponges, both asexual and sexual. We see this in multiple invertebrates. We actually see this in a lot of, we've seen this a lot already. Let's fix that word. Generation, there we go. Okay, so several types of asexual reproduction. Budding, we've seen budding before, like yeast, and we see budding in multiple invertebrate groups. We see regeneration as well. Regeneration is very impressive in these creatures. You can actually take a small chunk of sponge into the lab and really just damage it significantly, and then wait, and one tiny little fragment is left. That whole entire sponge can regenerate from that one fragment. So sponges are well known as maybe the most impressive regenerators. And then they also produce something called a gemule, which I would say is akin to some type of spore. We saw spores in bacteria and also in certain protists and fungi. And so the poriferans also have this survival mechanism where if all goes bad, a gemule can be produced, and that is a really hard spicule encased cell or series, a number of cells, and then that can survive the harsh conditions that might be occurring, and then when conditions change, can regrow into a new adult sponge. So budding, regeneration, and gemmules. 3 asexual means of reproduction. And then lastly, sexual reproduction in sponges. Like many marine invertebrates are broadcast spawners, right? So they're just shooting out massive numbers of gametes. They're overwhelming the predator population that way, higher likelihood of sperm and egg uniting. And different sponges do it differently. Some sponges will produce sperm and eggs, others will just produce one. Often, this is often the case, that the sperm will travel to the other sponge. Sperm from one sponge, quote unquote, a male sponge, will travel and will go through a pore and fertilize an egg in a female sponge. And then produced is this free-swimming larva. This is where we get back to a motile larva, but a sessile adult. And so this is your typical sexual reproduction in the sponges. That larva will be free swimming until it finds a hard substrate to settle on, and then it'll fix itself and start to mature. Okay, guys, that's our introduction to sponges. I hope you enjoy. And our third video of this week will be on phylum Cnidaria. And so this is going to be how I proceed. Is going to be one clip per phylum that we're going through. And I hope you enjoy this, and we'll see you next time.